Hello everybody! So in this video I'm going to show you how to complete a Punnett square. Punnett squares are used to show all of the possible offspring that can occur when two organisms with a known genotype mate. So of course we've covered phenotype and genotype, you know what those are now, so now we're going to apply those and see how those are useful to predicting the offspring of two individuals. So for our example, we are going to use pea plants. Of course, you know Gregor Mendel experimented with pea plants and he's the father of genetics, so we have to pay homage to him and use pea plants in our example. Mendel discovered that in pea plants there were dominant and recessive alleles for flower color. So he discovered that a flower with a white phenotype had a homozygous recessive genotype. I'm using R because you can easily distinguish between the letters. It doesn't really matter what letters you use to create your genotype. Gregor also discovered that flowers with a purple phenotype had two possible genotypes. They could either be, of course, homozygous dominant, or they could be carriers for the trait and could be heterozygous. So they can have one dominant allele and one recessive allele. Now, why is this important? What does that mean for us? So let's say that you're a florist and you have lots of purple pea plants, but you want some white pea plants too. So let's say that you take a purple pea plant and you know that this pea plant is homozygous dominant. And then you take one of your poor little white pea plants, and you know that white means that the genotype will be homozygous recessive. Now let's say that you want to figure out how many white flowers you're going to end up with. You're going to draw a square, and at the top of that square, first you're going to divide it into 4 sections. At the top of that square, you're going to put the genotype of one parent, doesn't matter which, normally it's the father, but in this case they're plants, doesn't really matter. And then you're going to put the genotype of the other plant on the side. Now you're going to do a little tic-tac-toe action here, and you are going to take the R and drop it down, and the other R and move it over. So in this box you're going to end up with a big R and a little r. And you're going to do the same thing here. You're going to drop this R down and you're going to move this R over. Same thing applies here. You're going to take this R and drop it down into this box, and this R and move it over, and then this R, drop it down to this box, this R, move it on over. So now you can see that all of our offspring, so 100% of our offspring are going to be heterozygous. So we're not going to end up with any white flowers at all in this cross.